Scientific and technical session with Chairman Dr. Avanath Ganguly and moderator Mr. Mrinal Kumar Bandyopadhyay. May I request Dr. Ganguly and Bandyopadhyay to come into the dais.
Of course. The same outfit you were wearing when Right, black.
Perfect.
Yeah. Black
Make
is
them
beauty.
so
Good morning, everybody. Am I audible?
Yes.
Yeah. So, I welcome you all to the first session of this morning. This is a scientific session, and in this session, our first speaker is Mr. Asim Poddar. May I request Sri Asim Poddar to present his lecture. Thank you.
হ্যালো, ঠিক আছে না? নমস্কার। Good morning, everybody. At the very outset, please, I beg your apology for this too long title, boring title. Okay, but I hope you will enjoy my talk. I would like to dedicate my talk to two renowned scientists of 19th century. One of them is Carl Abbe, who discovered Abbe diffraction limit of microscope in 1873. And another person, Carl Pearson, who formulated Pearson correlation coefficient in 1896. One, Carl Abbe, discovered the Abbe diffraction limit more than 150 years ago, and Carl Pearson formulated Pearson coefficient more than 125 years ago. Actually, today's my talk will focus on Abbe diffraction limit, Pearson correlation coefficient, in the context of modern digital photography. Life is beautiful. Life is beautiful and interesting, but the life process, it's really very, very complex and mysterious. Scientists actually there are several biochemical reactions are handled every time within a cell of living system. And to study the life process in depth, scientists are interested in photomicrography. What is photomicrography? We, as a photographer, we love our camera. Camera is an optical instrument. And if we add this optical instrument with another optical instrument, microscope, so we can take the picture of very minute objects. And photomicrography is the specialized domain of photography where images of minute specimens magnified by a microscope are recorded. You see a beautiful photomicrograph of plant root section. Actually, roots needs glucose. Glucose is formed in leaves by the method of photosynthesis, and then this glucose is transported into root because it needs energy, and that energy is required so that it can penetrate strongly into the mud, into the soil. Okay, can you see this glucose molecule here? Is it visible? Glucose molecule? No, it's not possible. It's not visible. Why? Why? Because it's not, it's present, but it's not visible. Why? Because you have to identify the molecule. No? So how you identify a specific molecule? How can a specific molecule be identified among the crowd of molecules inside a cell? Actually, fluorescence photomicrography could be a powerful tool to solve this problem. What is fluorescence? I won't enter into the physics too much. Actually, in our world, there are some materials which is known as fluorescent material. In short, is called fluorophore. And if you illuminate with some specific light, then it will glow with another light, another color of light. Okay? So this is the beauty of fluorescence. And if you tag this fluorescent molecule with your interest molecule, so then it will also glow. Okay? So by this way, you see, this is ordinary photomicrograph, and this is fluorescence photomicrograph. You see, in the fluorescence photomicrograph, everything is not illuminated. Okay? Only some part is highlighted. It's like a highlighter pen. It's very less, but it's very specific. So less, but less is better, because less but pinpointed, specific. Only the molecule present in this here is visible, but not these parts. No, it's black. Okay. You see, if you like to identify two molecules in your system, then you just label your material with two different dye, fluorescent dye, and you can see, here you can see it's a nucleus, okay? Nucleus. Can you imagine, can you guess what is the picture? Is it bird? No, it's not a picture of bird. It's a photomicrograph of a cancer cell, okay? Cancer cell. Okay. And this is nucleus. This is nucleus, blue color, okay? And this is actin filament. Actin filament is required to maintain the size and shape of a particular cell, okay? It's used as a scaffold. It protects its structure, okay? It's a protein, okay? And it's a cancer cell, and it's treated with some drugs, and you see the result. Nucleus is almost intact, but the actin filament, the scaffold, has destroyed. So eventually the cancer cell will die. Okay. So thing is that you see the power of fluorescence photomicrography. Just simply by showing your picture, actually we know, na? Seeing is believing. No, it's a very straight cut thing. And photograph is worth of 1,000 words. So instead of writing 1,000 words, just display your photograph. It will tell 1,000 words for you. No need to say 1,000 words in your thing. Okay. Oh, so many biochemical reactions are taking place every time in our cells. Okay? But this is very complicated. Okay? Forget about it. Actually, a very simple model of biochemical reaction can be expressed as A plus B. That is, if you A plus B is converted to C. Okay, it's a very simple type of model. Okay, but what is the criteria to be a positive reaction? A plus B must be in close contact if they take part in a biochemical reaction. Okay, A plus B. So then the A molecule and B molecule, they should come in a close contact. Otherwise, it's not possible. This is the primary requisite for a chemical reaction. And this is the okay, I already mentioned. You see, suppose there are two types of molecules. Okay, one green, another is red. And if they like to react, then they must find their pair. Otherwise, reaction would proceed. Okay. and that is called that type of colocalization. Colocalization means localization of two molecules can be photographed by colocalization photomicrography. Is a powerful tool to find the molecular colocalization. Procedure for colocalization is how it's proceed. First, two supposed interacting molecules are labeled with two different fluorophores. For instance, one produces green fluorescence and another produces red. And then two separate photomicrographic images are taken specifically for two individual fluorophores. You see, that is a two individual fluorophore. Okay, two individual photomicrographs. And then two images are marked by channel mixing and colocalization interference. Colocalization inference is drawn from observing the marked image. This is a primary image, one for green, another for red. And what would be the marked image? Looks like that, or it may looks like that. Why? Because you see here the difference between green and red is large enough, so they are not reacting. They are well apart. Okay, but here is yellow. That means the red and greens are very close in contact. But why is yellow? Because color change of color in the marked image indicates a clue of colocalization. That means the red and green molecules are in very close. They are in close contact, and that's why the color changes to yellow, because they share the same pixels of your picture. Okay, and why yellow? Because if red and green combines, then yellow is formed. In the additive method, all of we know as a student of photography. But here I have one question. The question is that it's a molecule. Molecule must have occupy some space, isn't it? Whatever the size and shape, it should take some space. Is it possible for another molecule to occupy the same space at the same time? Is it possible logically? No, it's not possible. If these two molecules do not share the same location, then why of their image share the same pixel? Is my question. The answer was given 150 years ago, more than 150 years ago, by Abbe. He discovered the Abbe diffraction limit to resolve. It has some limitation for resolving power. Okay, if it is too close, two molecules are too close, then I won't get one point. It will be blurry, one large point. Okay, we cannot distinguish two different points if they are in a very close system. So, is diffraction limit curse or blessing? Actually, it's not curse, it's a blessing. Why? Because if you take the green channel and red channel and then marks, then you will see if the molecules are very close in contact, then it will be represented as yellow. If these molecules, green molecules are alone, there is also red tint, because they are also alone. But most of the green and red molecules are interacting. That's why yellow is coming in your picture. Okay? So seeing is believing. Photography is equivalent to 1,000 words. So inference drawing from the masked image. If it is red and green separate, so no chance of interaction, because molecules are well apart. If it is yellow, then it's a clue of chance of interaction. Seeing is believing. Photograph is worth of 1,000 words. Is the saying seeing is believing always true? No, it's not always true. Why? If it is red and green, then it's fine, no chance of interaction. But if it is yellow, then we can't say absolutely that yes, the interaction is going on. The molecules are too close. Why? Because the Abbe formulated this equation, distance between 2 points that can be resolved is nothing but a lambda, that is wavelength of the light used by numerical aperture into T. Okay? I don't want to go detail, but thing is that the shorter wavelength, lambda means wavelength of light. Okay? Shorter the wavelength of light, smaller the resolving distance, that means better resolution. So violet light, very small wavelength, okay? So resolution is higher, okay? Good resolution. And by calculating, we can achieve that 200 nanometer. That means 200 nanometer is the distance, minimum distance, is a better, at the best resolving power is 200 nanometer, okay? If we take the photograph in violet light, okay? So you see, spatial distance between two molecules is 500 nanometer, then you get that type of picture, red and green, separate. If it is 100 nanometer, then it will receive that type of yellow picture, okay? And if it is 5 nanometer, then we'll get yellow, okay? If the distance between two molecules is 5 nanometer, then molecular interaction is possible. But if it is 100 nanometer, then molecular interaction is not possible. 100 nanometer is too much. It's not possible to react. So both 100 nanometer and 5 nanometer give the same type of result. So by seeing this result, you can't predict that yes, molecular interaction is going on, isn't it? So, is it possible to see beyond the diffraction limit? Is it possible? Seeing without seeing is possible. Seeing without seeing. Seeing through the mind's eye. Actually, seeing without seeing is possible by establishing a correlation between the concentration of two molecules. Concentration of two molecules. You see, red and green, two type of molecules are there. Okay, sorry. Apparently, it looks haphazard. Okay, but there is a correlation. You just take 1, 2, 3, 4 small circles. Okay, and count red and green. In circle 1, 1, 1. In circle 2, 1, 2, 3, 4, 1, 2, 3, 4, 4. In circle 3, 0, 0. In circle 4, 2, 2. So there is correlation. Increasing, when the red molecule is increased, green molecule is also increased. When red molecule decreased, green molecule also decreased, isn't it? So it's a case of correlation. But here, that type of correlation is not visible. Okay? In 1, only red, no 0, no green. In 2, 2 green, only 1 red. So it's not a case of relation. There is no correlation. Okay? So that type of info, it's a very schematic drawing. So it's possible to find if there is a correlation, you can find. But in real situation, just by viewing a complex photomicrograph, it's not possible to conclude that is there any correlation or not. So then you have to take the advantage of statistics. Pearson correlation coefficient, that is formulated in 1896. PCC, that is Pearson correlation coefficient, is used to find a correlation in general. In fluorescence photomicrography, it can be used to establish a correlation between the concentration of two molecules. PCC is statistical image analysis system based on pixel by pixel analysis of digital image. Oh, it's a complex formula. Okay, but it's not so complex. Only it has three variables. Total number of pixels in your image is very easy to find out. And fluorescent intensity value in one channel, say the green channel, that is also available. And corresponding intensity value in the other channel, say red channel. Okay. So as the digital photography, your image data is stored in a digital form. So it's just numerical data. So no problem. Just for calculation, raw materials supplied by the digital photography, no? And so suppose your image bit depth is 8. So intensity value of a pixel would be any number between 0 and 255. Suppose there are 1 million pixels in each channel. Then you just make one table. So pixel number 1, what is the green channel intensity? What is the red channel intensity? Pixel number 2, what is the green channel? What is the red channel? 1 millionth number of pixels, no? What is the green channel? What is the red channel? Figure, intensity value. And then just calculate according to your equation. And now software is available. And result would be, result lies between -1 to +1. Okay? Within that limit only. Okay? And 0 or 2, any value, if you yield 0 or near 0, then it's a region of weak correlation. That is, almost there is no chance of correlation, okay? No chance of interactivity. And if it is value +1, the region of strong positive correlation. Again, if it is the value towards negative -1, so it's a region of strong negative correlation. Okay. So you see, red channel, green channel, masked image. The Pearson correlation is 0.8830. See another example. Here you see the correlation is 0.76. That means this is much more correlated than this one. Okay, and you see the picture. Okay, so what is the role of digital photography in fluorescence photomicrography? First role is seeing is believing. Okay, and second, seeing with mind's eye, that is with statistics, like PCC is 0.8830. It's like that. So actually here, digital photography plays dual role. One is seeing is believing, and another is statistics. In this juncture, I like to quote Prashanta Chandra Mahanavi's once upon a time he said that seeing is believing, but statistics is truth. So, Abbe diffraction limit was discovered in 1873. Pearson correlation coefficient is formulated in 1896, but we couldn't use this Pearson correlation coefficient to overcome this Abbe diffraction limit, because at that time digital photography was not in picture. With the advent of digital photography, this is possible. Okay? Thank you very much.
Thank you very much, Mr. Asim Poddar, for your illuminating talk. If anybody has any question or discussion you would like to have with Mr. Poddar, please stand up and speak out. Thank you. If there is nobody, then I must thank Mr. Poddar for his very illuminating talk and enriching us with a great idea of the use of fluorescence photography in this field of genetics or photo. So I thank you once again, Mr. Poddar.